Hello class, welcome back. In this lecture video, we're going to discuss the difference between passive transport and active transport. So where we left off before the holidays, where we were discussing about the concept of passive transport, passive requiring no ATP energy at all to allow diffusion to occur across a selectively permeable membrane. There were two types of passive transport that we had discussed. We had what was called simple passive diffusion or transport, and then facilitated transport or facilitated passive transport. They're all the same thing. With simple, your solute can go directly through the lipid bilayer and pass through the phospholipids. To do that, these molecules have to be very small as well as non-polar, so they can cross this hydrophobic tail region. Or if molecules are much larger, so they cannot fit through the lipid bilayer, or they have a charge, so that means they're either polar or ionic, they will require an integral membrane protein to help them get through the lipid bilayer, either through an open channel or a carrier protein that does one or two molecules at a time rather than just being open for anything to flow through. But again, either one, simple or facilitated, is going to be high to low concentration and require no ATP energy. So our focus in this lecture is going to be the following. All right, for active transport, this time it's going to be the opposite direction. No longer is it going to be from high to low, but in fact it's going to be from low to high. So not high to low, it's going to be low to high concentration. To do this though, it's going to require energy. All right, and besides ATP energy, you will definitely always need the help of a transport or integral membrane protein for this to occur. So down below in the diagram, again, you can see both simple transport as well as facilitated transport, going directly through the lipid bilayer or going through a carrier protein. Again, high to low, you do not see the expenditure of any ATP energy, but now down below here, notice the arrow direction, correct? Alright, so you're going to be going from low to high this time. This picture happens to be showing you going from the outside of the cell to the inside, but it could also be the opposite direction. It could be from inside to out, as long as you're going from low to high concentration. Alright, you can see that you're using a carrier protein to do this, as well as you now have the expenditure of energy in the form of ATP. So you can see ATP being used here, but you did not see that up here with facilitated diffusion. So the only similarity that you really would see between active transport and facilitated transport is both transport mechanisms need an integral membrane protein, but that's where the similarities end. Alright, after that, active transport once again is going to be low to high, and it's going to require ATP energy to do this. One example of active transport, there's tons of different examples. We're going to focus on one of the most commonly taught ones, and that is the sodium-potassium pump. Now, with the sodium-potassium pump, you're going to use an integral membrane protein to help pump sodium ions against their gradient, as well as potassium ions against their gradient across the lipid bilayer. So the purpose of this is ultimately to keep water levels inside a cell balanced. So you don't want to have like 90% water on one side and only 10% on the other. You're trying to maintain an equilibrium, a 50-50 balance of water on both sides of the membrane. That way your cells won't shrink or expand too much. Another reason for this would be you create a charge on both sides of the membrane, both outside and inside. All right, what you're actually going to create, I'll show you in a second, is a positive charge, both inside and outside. That means you're always going to be attracting anything that has a negative charge, which is really important to help maintain blood pressure in your veins and arteries, as well as your heart muscles contracting, particularly your cardiac ones, of course, right? So your heart keeps pumping in and out, because that electrical signal that's being transmitted through your nerves is always going to be very easily conducted from both the outside and inside of the cells because of that positive charge that's being created. Alright, so for example, to show you here in this GIF, alright, we have both just a standard picture, but then we have the GIF over here. So ATP was just used to help pump sodium ions to the outside of the cell. And now you can see that potassiums are coming in too. So here's that ATP being used to help pump 3 sodium ions outside And as soon as these 3 sodium ions are pumped to the outside, another couple potassiums sneak their way inside as well. So you're always creating a gradient, an artificial gradient of a lot of sodium ions outside, and then you're always helping to maintain a higher concentration, in this instance the green ones, which would be potassium ions, inside. So you have a high positive charge as well as a high positive charge inside and out, which is going to allow the conduction of electricity or an electrical current. Electrons, again, are negatively charged both across the membrane really quickly. So if I just go back to refresh it, here comes 3 sodiums. I need ATP to help change that integral membrane protein structure to release the sodiums outside, and here comes 2 potassiums sneaking back inside. They kick off that one phosphate group. Then another ATP will come in, and you can see how that just keeps going in and out to maintain these exorbitantly high concentrations of both sodium and potassium ions. Notice that once ATP is used, you end up with something that's called ADP. This is like an uncharged battery. So if you got a new PlayStation or Xbox for the holidays, and your remote starts to die after a while, you need to put some new batteries in or a new rechargeable battery. Well, these ADP molecules can be recharged, just like you would plug a battery back into a charger. They're going to be recharged by the food that you eat. So when you break down your food, the breakdown of your food, in particular glucose, helps to add a third phosphate group back onto ADP, D stands for di, to create ATP over here again. So again, one example of why you would want to pump against the gradient is the sodium-potassium pump, creating an artificial positive charge on each side of the membrane so that you can get a decent electrical conductivity across here, especially in your heart muscles. Now, another way that active transport can be used is in a larger concept that we call bulk transport. Bulk means a lot of material, right, at once. So for bulk transport, we're going to be moving very large molecules or mass quantities of materials in or out of a cell. All right, now when you do this in large quantities, even if the flow is from high to low, if you look over here, when you actually have to open up the entire cell membrane here to take a big bite of something outside, or a big drink of water, think of it that way, to have a mass movement of the membrane like this is going to require ATP energy, regardless if the flow is high to low or low to high. So again, most times the materials that are being either brought into the cell or being released through the cell, we're going to be using vacuoles, which are going to be those larger membrane-bound structures for transport. And again, as I had mentioned, bulk transport is still considered a form of active transport since it will require ATP. So even if you do not see ATP expenditure, if we're not zooming in and looking at a cross-section of the lipid bilayer like we did in the previous pictures, all right, and we're just seeing the membrane opening up in massive quantities like you see here, you should automatically realize that's a form of bulk transport. So bulk transport is going to fit into 2 categories: endocytosis or exocytosis. Endo means in, so endocytosis is going to refer to large materials or amounts of materials being engulfed or brought into the cell membrane. So this could be large quantities of either solid or liquid. All right, so if the cell is low on water inside, it may take a big, huge gulp of water to bring that in at once. That's going to require energy. So endocytosis really gets broken down into 1 of 2 forms. You have what's called either phagocytosis or pinocytosis. If I refer to something as phagocytosis, again, it's endo coming in, but it's going to refer to solid particles. So maybe large bacterial cells that are being engulfed by a white blood cell to be broken down and destroyed. Versus pinocytosis, again, it's a form of endo, but this is when you take a big drink of water. So up here in this diagram, these blue dots could be either larger solid particles being engulfed, or it could represent water molecules being engulfed at the same time. And then exocytosis is going to be when you release large quantities of materials at once from the cell into the extracellular environment. There are no other breakdown terms for this one like we had for phago or pino. So regardless if it's solid or liquid being released from the cell this time, we simply call it exocytosis. So again, if we look here at this diagram, all right, both examples of endocytosis, both phago and pino. Again, bulk transport examples, both would require ATP energy. So here is phagocytosis. Here's a big, huge solid particle, maybe like I said earlier, a big microbe that's being engulfed by a white blood cell. The cell membrane actually has to reach out and give it a big hug and engulf it and bring it inside of the cell into a large food vacuole. Alright, so this is a big, huge item that was still way too big to come through an integral membrane protein, so you had to expend a little ATP energy to reach out and bring it in. As well as if you need some more extracellular fluid, or maybe there is salt that the cell needs that's dissolved in the water that's outside, it's going to take a big, huge gulp of water and bring it into the cell, into another small vacuole or large vesicle. It really depends on ultimately the size of and how much volume of material is being brought in. But regardless, phago going in is a solid particle, pino going in is going to be large quantities of liquid. And to finish out, we can see both endocytosis and exocytosis in action. So once again, here is a microbe or some other large food particle that is being engulfed by, in this example, we do have a white blood cell. So it engulfs this microbe. Let's say it's something like E. coli that can make you very sick. All right, next you're going to have one of our good old boys here, the lysosomes, which have digestive enzymes in them. We had learned that they break down both waste and food particles. Well, this bacterial cell is going to be considered waste, something that we don't need. So once it is engulfed via phagocytosis into a large vesicle or a small vacuole, a lysosome would fuse with it, break it down and digest it, and whatever the cell doesn't need then, the waste products, will then be released in bulk via exocytosis to the outside environment. So all this debris and waste product that you're releasing then from the breakdown of that microbe, if this was your body, it would be released eventually into your blood. This waste would be filtered out by your kidneys, and eventually once you urinate, you would also be releasing some of this waste product that was destroyed by your white blood cells. Again, all this is being done by all of your cells in order to maintain homeostasis. So in conclusion, we just learned about active transport, which in general is transport from low to high concentration and requires the use of ATP energy. Or if we discuss bulk transport, the movement of either large solid or liquid materials to the inside of the cell or the release of large amounts of material at once, they would both require energy or ATP as well, so they would still be considered active transport even if they happen to be going from high to low concentration. If you have any questions, be sure to ask me during our class period. Thank you.